Every time your skeletal muscle contracts against resistance, it acts less like a simple mechanical lever and more like an endocrine organ, pumping out a cascade of potent biochemical signals that fundamentally alter your body's internal state. For decades, we viewed muscle tissue merely as the engine of movement, something we train for strength or aesthetics. But modern molecular biology has revealed a far more profound truth. Your muscles are actually your body's premier pharmacy. When you contract them, they manufacture and secrete powerful protein messengers called myokines. These molecules travel through your bloodstream, crossing the blood-brain barrier, reaching your vital organs, and triggering immediate anti-inflammatory and cellular repair pathways. If you want to slow down biological aging, understanding and activating this muscular signaling system is one of the most powerful tools at your disposal. To understand why myokines are so transformative, we first need to look at the silent destroyer of healthspan, chronic low-grade inflammation. Scientists often call this inflammaging. Unlike acute inflammation, which is your immune system rushing to heal a cut or fight off a sudden infection, chronic inflammation is a slow, smoldering fire that persists for years, gradually wearing down your tissues, damaging your DNA, and accelerating cellular decay. It is driven largely by fat tissue, particularly visceral fat around your organs, which pumps out inflammatory signaling proteins called cytokines. These rogue cytokines signal your immune system to stay on high alert, creating a hostile environment inside your body that contributes to cardiovascular disease, metabolic dysfunction, and cognitive decline. For a long time, we thought of this as an inevitable consequence of getting older, a tax we pay for simply living. This is where your skeletal muscle steps in as the ultimate countermeasure. Skeletal muscle is the largest organ mass in the human body, typically making up about 40% of your total weight. When you engage in physical activity, your muscle fibers undergo mechanical stress, leading to microscopic tears and the activation of signaling pathways within the muscle cells themselves. In response to this workload, muscle cells dramatically upregulate the production of myokines. The most famous and thoroughly researched of these is interleukin-6, or Illinois-6. Now, in the context of fat tissue or chronic disease, Illinois-6 is inflammatory. But when Illinois-6 is released directly by contracting muscle fibers, it acts entirely differently. It becomes an anti-inflammatory powerhouse. Muscle-derived Illinois-6 acts like a biological flare, signaling your immune system to stand down and actively suppressing those destructive inflammatory cytokines. Think of muscle-derived Illinois-6 as a master coordinator of your body's internal repair crew. Once released into the circulation, it travels directly to your liver, stimulating the production of anti-inflammatory compounds like interleukin-10 and interleukin-1 receptor antagonist. At the same time, it dampens the production of C-reactive protein, a primary clinical marker of systemic inflammation. By simply contracting your muscles, you are chemically interrupting the inflammatory cascade at its source. But Illinois-6 is just the opening act. The moment your muscle fibers start firing, they trigger a cascade of secondary myokines, each with specialized duties designed to protect your brain, optimize your metabolism, and renew your cellular machinery. One of the most fascinating myokines discovered in recent years is Irisin. Named after Iris, the Greek goddess who acts as a messenger of the gods, this molecule is manufactured when muscle contraction activates a specific transcriptional coactivator called PGC1-alpha. Irisin is essentially a metabolic conductor. Once released, it travels to your white fat tissue, the stubborn fat that stores excess energy and drives inflammation, and forces it to undergo a process called browning. White fat cells are lazy and storage-oriented, but brown and beige fat cells are metabolic powerhouses packed with mitochondria, the tiny cellular generators that burn fuel for heat and energy. Irisin essentially reprograms your fat cells to start burning energy rather than storing it, dramatically improving your metabolic flexibility and insulin sensitivity. Irisin does not stop at your fat tissue. It also crosses the blood-brain barrier and triggers the production of brain-derived neurotrophic factor, or BDNF. You can think of BDNF as high-octane fertilizer for your brain. It stimulates neurogenesis, the birth of new neurons, and strengthens the synaptic connections between existing brain cells, particularly in the hippocampus, the region responsible for memory and learning. When you contract your muscle, you are literally bathing your brain in a chemical environment that promotes neuroplasticity, guards against cognitive decline, and clears out cellular debris. Let us look at the data to understand the scale of this impact. When we measure systemic inflammation markers in individuals who engage in regular muscle contraction protocols compared to sedentary controls, the contrast is staggering. As you can see in this chart tracking circulating inflammatory cytokines relative to weekly muscle loading sessions, the reduction is profound. The data clearly demonstrates that as you increase the frequency and quality of muscle contractions, inflammatory markers drop exponentially. This is not about vanity or athletic prowess. This is about keeping your internal biological terrain clean, pristine, and resilient against the ravages of time. Beyond myokines, muscle contraction initiates deep cellular repair through another critical mechanism, autophagy. Think of autophagy as your cell's internal recycling and cleanup crew. As your cells go about their daily business, they accumulate damaged proteins, oxidized lipids, and worn-out mitochondria that produce harmful free radicals. If left unchecked, this cellular garbage builds up, causing cellular dysfunction and premature aging. When your muscles contract intensely, they experience a temporary energy deficit and mechanical stress. This activates an enzyme called AMPK, which acts as the cellular master fuel gauge. AMPK sounds the alarm, shutting down growth pathways and turning on autophagy, instructing your cells to sweep up the damaged debris, break it down into basic amino acids, and repurpose them for new, healthy structures. This is precisely why physical inactivity is so biologically toxic. When you sit for prolonged periods, your muscles remain dormant. The myocrine machinery turns off. The anti-inflammatory signals cease, autophagy slows to a crawl, and the inflammatory cytokines begin to accumulate. You are essentially starving your body of the essential biochemical messages it evolved to expect every single day. To harness this pathway effectively, you need a precise strategy. Enter the True Diagnostic Test, the definitive solution to measuring biological age and tracking your internal rate of aging. While we can easily measure how many years have passed since our birth, chronological age tells us very little about our actual cellular health. The True Diagnostic Test analyzes your DNA methylation patterns using advanced epigenetic clocks, most notably the Dunedin-Pace algorithm. Unlike older clocks that simply estimated your accumulated wear and tear, Dunedin-Pace acts like a speedometer for your aging process, measuring the rate at which your biological system is deteriorating right now in real time. By implementing targeted muscle contraction protocols and testing your methylation status over time, you can scientifically verify whether your interventions are actually slowing down your biological clock. So how do we turn this science into a daily protocol? Let us build the myokine protocol. The first pillar is mechanical tension through progressive resistance training. To trigger a robust release of myokines like Ilion-6 and Irisin, your muscle fibers must experience significant mechanical load. This means lifting weights, using resistance bands, or performing rigorous bodyweight movements that challenge your strength. You do not need to spend hours in the gym. 2 to 4 sessions per week, lasting 30 to 45 minutes, focusing on compound multi-joint movements like squats, deadlifts, presses, and rows, is entirely sufficient to activate the signaling pathways. The second pillar is movement frequency throughout the day. Even if you hit the gym for an intense workout in the morning, sitting for the remaining 10 hours of the day dampens your overall metabolic health. To combat this, introduce movement snacks. Every 45 to 60 minutes, stand up and perform 2 minutes of active muscle contraction, bodyweight squats, wall sits, or brisk walking. This brief contraction keeps your muscles active, maintains baseline myokine production, and prevents the cellular stagnation associated with prolonged sitting. The third pillar is intensity modulation through Zone 2 and Zone 5 cardiovascular exercise. While resistance training provides the mechanical tension, steady-state aerobic work and high-intensity intervals recruit different muscle fiber types and enhance mitochondrial biogenesis. Combining resistance training with cardiovascular work ensures that your entire muscular system is continuously producing this rich array of excitokines and myokines. Let us look at how these interventions compound over a multi-month protocol. As your muscle mass increases and your myokine output stabilizes, your body undergoes a profound shift in systemic homeostasis. Let us review the data tracking improvements in metabolic health markers over a 12-week targeted myokine protocol. The numbers speak for themselves. You have direct, sovereign control over your biology. Every time you contract your muscles with intention, you are choosing to flood your system with anti-inflammatory protectors, brain-boosting neurotrophins, and cellular renewal signals. Your muscles are not just tools for movement, they are your absolute best defense against aging. If you want to dive deeper into how specific protocols interact with your cellular machinery, click on the video showing on your screen right now to discover how optimizing your mitochondrial function can take your longevity strategy to the next level.